My name is Brotman. I'm a graduate student working with Ralph Adolphs at the California Institute of Technology and Uli Rudishauser at Cedars-Sinai Medical Center in Los Angeles. I might have started that a little early. Sorry for the timing. But what I'm going to be talking about today is a new project that we've been working on trying to investigate the single neuron correlates of trait judgment. And I really want to narrow this down to a very specific kind of trait judgment. It's not just any trait judgment, but the kind of automatic, almost gut feeling that you get the first time that you see someone, sort of automatic assessment that you make about their character. Not to say that there's anything, there's no ground truth on this. But to kind of ground us in the kind of judgment that I'm talking about, I'm going to put a couple faces up on the screen, and I want you to think about which of these people you think is warmer in their character. And I really don't have to leave this up for very long at all to feel pretty confident that you would say that it's the person on the right. And I want to use this to highlight a couple of the things that we find really interesting about this kind of judgment. The first is that it's almost instantaneous, so you make it within the first second after you see a face for the first time, and it's pretty ubiquitous. So you make it almost every time you see a face. And to kind of give an idea for how we're looking at this problem, I'm going to work through some of the background on the social psychology and the neuroscience of this topic. Then I'm going to talk a little bit about our recording setup and the task that we're using with our patients. I'm going to talk a little bit about the behavior that we see both in our subjects, but also in a larger group of behavioral subjects. And finally, the neural data that I think we're all really interested in and what we're seeing in that. And obviously, we're not the first people to be interested in this particular question. There's a very long history of social psychology literature trying to understand what kinds of judgments do we make about faces, what predict them. And there's some pretty robust results from this. And one of these is that while we can describe a face with a very, very large number of traits, if you ask a large group of people to rate all of these faces on traits from friendly to warm to competent to aggressive, what have you. We can reduce this to a relatively low dimensional space. And the number of dimensions varies, but usually from 2 to 4. In our case, we use a 4-dimensional parameterization for the stimuli that we show our subjects. This includes warmth, competence, femininity, and youthfulness. And to kind of give you an example of how we might parameterize one of these faces, the face on the left probably falls high on the warmth dimension, competency dimension, youthfulness dimension, and low on the femininity dimension. There are a couple other interesting features about the sorts of judgments that people make. And that's that they're consistent across groups of individuals. You take 2 large groups of people and ask them to rate the same faces. They come up with very similar ratings for those faces. The second is that a lot of these judgments, at the group average level at least, are highly predictable just by the features that we're able to parameterize from the face. On the neuroscience side, the research is a lot sparser. We have some indication from John Freeman's work that the medial temporal gyrus and the amygdala are both implicated in this sort of decision-making process. And on the single unit side, we know that there are neurons in the hippocampus and the amygdala that seem to be predictive of this sort of trait judgment. But when we compare it to something like face processa, trying to identify, how we identify a person in the fusiform gyrus or something like that, the literature is significantly sparser. And so this made us interested in whether or not we could try and leverage the spatial and temporal resolution that we get from single neuron recordings and use the access that we have to multiple locations in the face processing stream to try and understand how information about these traits changes as you progress from the ventral temporal cortex to the medial temporal lobe. And so now to dive a little bit into our recording setup, we do all of our recording at Cedars-Sinai. It's a collaborative effort between Uli Rudishauser on the left and our surgeon Adam on the right. And all the patients that we work with have pharmacologically intractable epilepsy. They're about to go undergo surgery. And before that, we use Benke-free depth electrodes to try and understand where in their brain the seizure activity is originating. At the very tip of these electrodes, we have a bundle of microwires, and each of these microwires is capable of recording an extracellular voltage trace from which we're able to isolate single neurons. And examples of these waveforms are above right there. And we are able to collect these recordings from a pretty large number of regions, but the ones that we're going to be interested in are the middle and bottom frames on the right, the hippocampus, amygdala, and ventral temporal cortex. And while we have these implants in, we record a task with our patients. There's a collaborative effort to build this task. But roughly how it's structured is that at the beginning of a block, we show our participants the definition of a trait, and we tell them they're going to be rating a series of faces on this particular trait. And then each of the faces that we show them, 20 per block, ends up being a fixation cross, just the face for about a second, and then we show them a rating screen, and we ask them to provide a rating 1 to 7 on a Likert scale of how much they think that face embodies that trait, the person embodies that trait. This ranges from not at all to extremely for each one of the traits. We do this for 4 of the traits, the 4 that I talked about in the parameterization earlier: warmth, competency, femininity, and youthfulness. And accompanying that, we also have a one-back task that we show another set of faces. In total, it ends up being about 360 faces, and all of them are novel. The patients are not familiar with any of them prior to the task. And the behavior that I'm going to talk about, actually, because these ratings can be so stochastic, is from a larger group of participants that we collected only behavioral data. We collected it from about 600 of them on Prolific. We showed them all of the same faces that we show our epilepsy patients. And so we're able to compile a group average rating for each one of these faces for each one of our traits. And right here I'm showing the distribution of each of these. There's a decent amount of spread on each of the traits. We see some of the features that we would expect. Femininity is bimodal, and the other ones are unimodal with a certain amount of spread there. And what's maybe more important is that we're able to lay out each of our faces. We can scatter them. And in this example, I'm showing you warmth on the x-axis and competency on the y-axis. And we see a similar pattern when we plot any of these 2 traits together. None of them are especially redundant. And that's really useful for the parameterization that we're interested in. Before moving on, it's probably worth noting, when we do this, we see the same features that we do in the social psychology literature. So 2 subsets of this large set of behavioral subjects that we collect. There's a consensus between them in terms of how they rate these faces. And with our epilepsy subjects, what we see is that they are, we show the same subject, the same face twice, their ratings correlate pretty well. And not just that, but their ratings correlate decently well with the group average ratings. So we're pretty confident that the subjects are actually engaged in judging these faces as they're sitting there, and that the judgments they're making are reliable. And so one of the neurons that I think we're actually most interested in, we can take these group average ratings and we can split them and try and understand, are the faces that rank highly on a particular trait represented differently by any of these individual neurons than those that rank low on any of those traits. And so we make the split, the top 50th percentile and the bottom 50th percentile in orange and blue. For this particular neuron from the amygdala, on the femininity trait, we see that yes, in fact, this neuron's activity does track really well, this highly feminine to low feminine split. And this is a particularly dramatic example. But we see a similar pattern if we split another neuron, also from the amygdala on another subject, dividing the highly youthful and the low youthful subjects. And on a population level, we've collected from 8 patients so far, totaling roughly 250 neurons in the amygdala and about 170 in the ventral temporal cortex. Of those, we find that a little bit under 50 in each area are well-tuned to some of the task features that we're interested in. Those you can see in blue on the right. This works out to somewhere between 10 and 15% of the amygdala neurons and roughly 25% of the ventral temporal cortex neurons. So the next thing that we were interested in was whether or not this separation based on trait ratings was robustly decodable at the population level. So what we did was we aggregated the neurons from each of our subjects into a pseudopopulation, and we attempted to train a linear decoder to distinguish whether they were high on a particular trait dimension or low on a particular trait dimension. And we did this for a set of our recording areas, the hippocampus, amygdala, and ventral temporal cortex. I won't be showing all of those today, because there was a particular criteria of robustness that we were interested in, and that was that we ended up at the same conclusion from this decoding analysis when we held out to any one of the subjects. And the reason for that was that we wanted to be sure that the results that we were getting were not the result of something idiosyncratic about one of our subjects. And so again, in total, we had 8 subjects and about 170 neurons in the ventral temporal cortex and about 250 in the amygdala. And so what I'm showing first here are the results from our attempts to decode femininity. The ventral temporal cortex on the bottom and in the amygdala on the top. And each of the dots that you're seeing on there is a decoder trained on a roughly 800 millisecond bin. Kind of unsurprisingly, we see that we're able to distinguish well between high and low femininity conditions. This wasn't particularly surprising to us. What was, however, was that when we attempt to do the same for youthfulness, we're able to decode this particular trait in the amygdala, but not in the ventral temporal cortex. Now, this was kind of surprising to us. This was largely surprising to us because from the non-human primate work, and also from some of the work that's come out of our lab recently, we know that the neurons in the ventral temporal cortex that we're recording from encode specific combinations of facial features that are the same facial features the psychology literature tells us are predictive of these traits. And so why this split wouldn't be decodable in the ventral temporal cortex was a bit puzzling to us, especially since it was later in the hierarchy in the amygdala. And unfortunately, I'm not going to be able to resolve that mystery today. It is, however, the question that's guiding the rest of the work that we're doing on this project. And we think that there are a couple not mutually exclusive reasons that this could be the case. One of them is that the youthfulness-related facial features, they're just more prevalent in the amygdala. More of the cells are tuned to those particular dimensions of the face space. Alternately, but again, not exclusively, it's possible that the amygdala is encoding more abstract features about these faces, something that requires a couple more layers of processing. And we're hoping, not just to collect a little bit more data, we're hoping that would give us more insight, but also to fit encoding models to the activity of each of these cells to try and predict their individual firing rates. And the way we're looking at doing that, the way we've been doing it so far in collaboration with a colleague of mine, is by taking visually selective cells and parameterizing each of the faces that we show the subject, in this case extracting the social features, the trait ratings that I talked about earlier from our group average, and also extracting some structural features, both from deep neural networks, but also from landmark placement on each of these faces. And once we fit linear models to predict the firing rates from these parameterized features, we can end up finding things like, in this case, that this cell's firing rate is predicted well by the facial geometry of the images shown to the subjects, but not by the social trait ratings. And if we look a little bit closer at the cell, we're able to find, in particular, which of the features drives firing in this cell. And on a larger scale, we're hoping to use this to try and understand what the distribution of facial features looks like as it's represented in both the the ventral temporal cortex and the amygdala, to try and give us a little bit of insight about what kinds of different information is represented in each of these areas. Hopefully that'll get us a little bit closer to an answer about, why is this youthfulness information represented in the amygdala but not the ventral temporal cortex. Like I said, really highly collaborative project. There are many, many people to thank, our funders as well. But with that, I'll close. And if anybody has any questions, I'm happy to answer them now. Or feel free to reach out at the email on the slide.
Thank you so much. That was fascinating. Just to say, I am checking the Q&A thing. So if you want to post questions there, you can do that as well. I had a quick question about, I like how you broke it down into sensory features, but have you looked at things like attractiveness or anything that's socially relevant, because the amygdala seems to care about those things?
Yeah. So we've done a little bit of that. So those were some of the ratings that we collected before. There were a series of dimensions that I didn't talk about that we were interested in as well, and we don't seem to see an especially strong effect for any of those either. I believe attractiveness was included in that, as well as aspiring and happy, and a collection of others that are escaping me at the moment.
Sure. And obviously the group ratings might not be ideal for that individual in terms of things like that.
That's another. We're particularly interested in the individual differences. What I will say from anecdotal experience is that many of our subjects will tell us, I can't make a judgment like this, and it'll seem like their particular ratings are, poorly correlated with the group average relative to other subjects. That doesn't seem to impair our ability to decode from their neural data.
Gotcha. Go ahead. You might have to turn that on. There's a switch.
Let's try that. Can you comment at all in terms of which subnuclei of the amygdala you're in, and whether there are any effects that are connected to that?
I can't, to be perfectly honest. I have not tried to determine whether or not this is driven by particular locations in the amygdala.
Do you think it is possible to
So the way that we localize these electrodes is we co-register a post-operative CT with the electrodes implanted with a preoperative T1. And there are atlases that subdivide the amygdala, and we can rely a little bit on the surgeon's labeling. So I know that other research from our group has attempted to do this with both the amygdala and the hippocampus with some success. It's just not something that I've done for our data yet.
If you stick around to the last talk, Denise is going to talk about segmentations of the amygdala. Anybody else got any questions? Or from the panel, you're free to ask as well. We have a minute.
Nice talk. It's nice to see your data. Can you tell us a little bit more about the time course of decoding? Did you say it didn't evolve until 800 milliseconds later? That sounds long, a long time.
So, not that it didn't evolve until 800 milliseconds later, but the decoding windows that we used were each 800 milliseconds long. Mm-hmm. And when you apply a sliding window like that, it seems to peak at the roughly 200 milliseconds to 1,000 milliseconds after image onset.
Great. Thank you, Austin.